Astianthus. Astianthus is a monotypic genus of flowering plants in the Bignoniaceae family. The sole species is Astianthus viminalis. It is known by the common names achuchil in Mexico and chilca in Guatemala and Honduras. Astianthus is native to Mesoamerica, from southern Mexico to Nicaragua. It is a shrub, or if larger, a tree of varying height, occurring mostly along streambanks from sea level to 1,200 and 3,900 ft in elevation. Its leaves are unusually long and slender, resembling those of a willow, possibly because it is a rheophyte. Description: the following description is excerpted from one that appeared in Flora Neotropica. Trees or shrubs, 2–20 tall. Leaves in whorls of 3, simple, linear, 6–30 cm long and 0.3–1.4 cm wide. Midrib prominent. Secondaries numerous, strongly ascending. Inflorescences terminal, paniculate, the lateral branches in whorls of 3. Calyx campanulate, 5-dentate, the teeth 1–3 long. Corolla yellow, 4–5.5 cm long, tubular to infundibuliform, base of corolla narrowly tubular. Stamens didynamis. Gynoecium 2.53 cm long, the ovary glabrous, its base surrounded by a nectariferous disc. Fruit fusiform terete, glabrous, 3–8 cm long, the septum perpendicular to a false septum, the combined structure cruciform in cross-section. Seeds small, borne perpendicular to the septum and parallel to the false septum, the seed body sharply demarcated from the wing. Taxonomy: the genus Astianthus was erected by David Donne in 1823 in the Edinburgh Philosophical Journal. Donne named its sole species Astianthus longifolia. He was apparently unaware that Carl Sigismund Kunth had previously named this species Bignonia viminalis in 1819. The authority for this name is often cited as HBK instead of Kunth. It is not clear why Owen Howard Gentry gives the date of the name as 1819. William Hemsley transferred this species to Tacoma as T. viminalis in 1882, recognizing that the specific epithet of Kunth had priority over that of Donne by the rules of botanical nomenclature. The name Astianthus viminalis is often accredited to Henry Ernest Balin in Volume 10 of his Histoire des Plantes, but the reason for the citation is not self-evident upon viewing page 44 of this work. Older works have usually placed Astianthus in the tribe Phytochemistry, but the circumscription of that tribe was greatly revised in 2009. Astianthus is now usually placed in Bignoniaceae and Certicetes. Owen Howard Gentry called Astianthus a very isolated genus with no obvious affinities, and further wrote that the superficial resemblance to Chelopsis is apparently due to parallel evolution for the same type of riparian site. Astianthus has not yet been sampled for DNA in a molecular phylogenetic study. Uses: where Astianthus approaches its largest size, useful lumber can be produced from it, but it is rarely harvested by lumberjacks. As with almost every plant, medicinal value has been alleged, but no verifiable evidence of efficacy has been observed. Isolated compounds and accrued extracts from Astianthus have failed to show any antimicrobial activity. They also showed no cytotoxicity for tumor cells. Phytochemistry: the pentacyclic triterpenoids ursulic acid and olenolic acid have been extracted from Astianthus. So have cinnamic acid, p-methoxycinnamic acid, and stigmasterol. A chloroform ethanol gradient elution high-performance liquid chromatography system was used to extract the iridoid glycosides campanoside and 5-hydroxycampanoside.